Manali, I hope you all are fine. In today's class, we are going to discuss about the digestion of proteins. In the last session, I already explained you about the digestion of carbohydrates. Now, let us learn about the digestion of proteins. Proteins are also the nutrients. These nutrients needs to be broken down into simple form so that it can be absorbed by our body. So, when you see in the buccal cavity, saliva is present. And in the saliva, certain enzymes are present that are responsible to break down certain substances. So here in the saliva, you cannot find any proteolytic enzymes. There will be no proteolytic enzymes present in saliva. So as the proteolytic enzymes are not there in the saliva, in the buccal cavity, no protein digestion takes place. So protein digestion cannot be seen in the buccal cavity. The digestion of proteins would not occur in the buccal cavity because of the absence of proteolytic enzymes. When you move to the other part of our alimentary canal, that is stomach, then in the stomach, certain enzymes are present that are in inactive form. Firstly, that enzymes should be converted into an active form. Firstly, they need to be present in active form. Then they can act on certain substances to break them in simple form so that it can be absorbed by our body. So what are that inactive enzymes? Let us see. In the stomach, certain enzymes are present that are in inactive state. So first enzyme is pepsinogen, which is present in inactive state. We all know that in stomach certain acids are present like hydrochloric acid. So that hydrochloric acid act on this pepsinogen and convert this pepsinogen into pepsin. So pepsinogen can be converted into an active form, that is into pepsin, by the action of hydrochloric acid. Once the pepsinogen gets converted into pepsin, now this pepsin which is present in active form, it can act on the proteins. It can break the proteins into peptones and also into proteases. So the proteins can be broken down into peptones and proteases by the action of pepsin. And pepsin is an enzyme that can act when the pH is 1.2 to 1.8. So the pH that is required by the enzyme pepsin is 1.2 to 1.8. One more enzyme is present in the stomach that helps in the digestion of proteins, but that enzyme is again present in inactive state, and that another enzyme is prorenin. So this prorenin can be converted into an active form by the action of pepsin. So pepsin is an enzyme that makes prorenin into an active form called renin. Now this renin is required to convert the milk protein into some simpler form so that it can be absorbed. So this renin acts on casein. Casein is a milk protein. Firstly, this casein is converted into paracasein, and then calcium is going to bind this paracasein protein. It becomes calcium paracasein. Now on this calcium paracasein, pepsin is going to act and it can be converted into peptones and proteases. So here both the enzymes are required to break the milk protein. Two enzymes are required that should be in active form. First is renin, second one is pepsin. Renin helps in converting casein to paracasein. Then this paracasein becomes calcium paracasein. Now on this calcium paracasein, pepsin is going to act, and by the action of pepsin, this calcium paracasein can broken down into peptones and proteases. So this is how the different enzymes like renin and pepsin, which are very essential for the digestion of proteins. So in the stomach, we need to always remember that two enzymes are essential, that are renin and pepsin. Both of these enzymes are required to break down different kinds of proteins that can be easily absorbed by our body. But pepsin is an enzyme that cannot break down some of the proteins like keratin. Keratin is a protein which is present in hairs or in nails. So in the different parts like hairs and nails, one protein is present, one protein is found, and the protein name is keratin. it cannot be broken down by the action of certain enzymes like pepsin. So pepsin is an enzyme that can only act on milk protein and some other kinds of proteins. So this is how the digestion of proteins takes place in the stomach. Next part of the alimentary tract is our duodenum. So duodenum is the first part of our small intestine, and we know that bile is present. Bile is transferred to the duodenum, but we have already discussed that bile is a juice which helps in the process of digestion, but it doesn't have any enzymes. So no enzymes are present in the bile, but in the duodenum you can find the presence of certain enzymes like trypsinogen. Trypsinogen is a substance that is present in inactive state. Again, trypsinogen can be converted into an active form, that is trypsin, by the action of an enzyme enterokinase. So enterokinase is an enzyme responsible to convert trypsinogen into trypsin. One more substance is present that is in inactive form. So once the trypsinogen gets converted into trypsin, this trypsin is required to convert chymotrypsinogen into chymotrypsin. So chymotrypsin, after it gets converted, both of the enzymes, trypsin and chymotrypsin, both of them are present in active state. Once the enzymes becomes active, these two enzymes, trypsin and chymotrypsin, both of them act on protein and break the protein into proteases and peptones again. So this is how the conversion of inactive enzymes into active enzymes takes place, and after the enzymes becomes active, these enzymes are required to break down the proteins into simpler forms like proteases and peptones. After the proteins gets converted into proteases and peptones, this cannot be easily absorbed. This needs to be converted into some other simple forms again. So these proteins and peptones, by the action of carboxypeptidases and aminopeptidases, they can be converted into amino acids and dipeptides. These dipeptides, again by the action of dipeptidases, can be broken down into amino acids. So amino acids are the small units. When you join these amino acids together, they can form a chain-like structure, that is protein. So proteins can be broken down into amino acids by the action of different enzymes that are found in the different parts of our alimentary canal. So this is how the digestion of proteins takes place in the different parts of the alimentary canal that is found in the human body. Let us move to the next one, that is digestion of fats. When we have food, we have the different kinds of nutrients like carbohydrates, vitamins, minerals, fats and proteins. So fats needs to be digested because they are present in triglycerides form, which needs to be converted into glycerol and fatty acids that can be absorbed by our blood. So let us see how these triglyceride or how these fats are getting converted into glycerol and fatty acids. When you see in the buccal cavity, then there are no enzymes present to break down the fat, so no fat digestion can be seen in the buccal cavity. But in the stomach, mild gastric lipases are present that can help to break down the fats, that can help in partial fat digestion. Not the proper fat digestion can be seen in our stomach. Negligible fat digestion, we can say. This negligible fat digestion occur in our stomach because of the inavailability of optimum pH. The pH which is present, the pH of the stomach is not suitable for the digestion of fats. So optimum pH is not available, so negligible fat digestion can be seen in the stomach. And the other reason is the fat cannot be emulsified in the stomach due to its pH. So these are some of the reasons by which the negligible fat digestion can be seen in the stomach of alimentary canal. But you move to the other organ, that is in the duodenum. So bile salts are the salts that can act on the fats, and these fats can get emulsified. It can be converted into emulsified fat. This emulsified fat, when the pancreatic lipase act on these emulsified fats, then they can be changes into diglycerides, and fatty acids. So in this way, the breakdown of the fats takes place by the action of different enzymes. So diglycerides and fatty acids are present, on which the lipase is an enzyme that acts. So by the action of an enzyme lipase, these diglycerides can be broken down into monoglyceride fat and fatty acids. Now these monoglyceride fats, again by the action of lipase, can get changes into glycerol and fatty acids. So this is how the breakdown of the fats takes place in the different parts of the alimentary tract. And finally, when the fats are broken down into glycerol and in the form of fatty acids, then they can be absorbed easily by the different organs of our body. Next one is nucleic acid digestion. So the digestion of nucleic acid is also one of the important aspects when we learn about the process of digestion. So digestion of the nucleic acid would not take place in the buccal cavity because of the absence of enzymes. Same in the case of stomach. Stomach is acidic in nature because the hydrochloric acid and some other acids are present. Gastric juice is present in the stomach. So it is acidic in nature and no nucleic acid digestion takes place in the stomach because of the absence of certain enzymes that are required for the breakdown of nucleic acid. But when you see the duodenum, then duodenum receives the pancreatic juice, and in the pancreatic juice some enzymes are present that are called as nucleases. Nucleases are of two types, deoxyribonuclease and ribonuclease. Both of them act on different substances to break them. Deoxyribonucleases are the enzymes that act on DNA, and this DNA can be converted into deoxyribonucleotides. Similarly, ribonucleases are the enzymes that act on RNA, and RNA can get converted into ribonucleotides. Now these nucleotides, again by the action of certain enzymes, can be changes into some more simpler form. Like nucleotides in the intestine, nucleotides are found like deoxyribonucleotides and ribonucleotides. So again the enzymes like nucleotidases. These nucleotidases are the enzymes that breaks the nucleotides into nucleosides and inorganic phosphate. These nucleosides, again by the action of enzymes like nucleosidases, they can be broken down into some pentose sugars and nitrogen bases like purines and pyrimidines. So in this way, the nucleic acid can break down by the action of different enzymes that are present in the different parts of the alimentary canal or digestive tract. So this is how the nutrient digestion, the different nutrients can get digested by the action of different enzymes that are found in the different parts of the digestive system. That's all for today.